Hello there. I'm so glad you found your way here tonight. Wherever you are in the world right now, allow yourself to settle into this shared moment of stillness. Let the noise of the day fade away. Let your thoughts grow quiet, like the gentle rhythm of your own heartbeat. Right now, as you listen to these words, something extraordinary is happening inside you. Billions of neurons are firing in intricate patterns. Electrical signals are racing through pathways you will never see. And somehow, from all that silent activity, the experience of being you emerges. Tonight, we journey inward, into the quiet landscape of the human body. A universe contained within your skin. A symphony of cells, chemicals, and currents that never stops playing. We will explore how your bones rebuild themselves in silence, how your eyes can distinguish millions of colors, how your heart beats over 100,000 times each day without ever being asked, all of it waiting to be whispered into the stillness. Tonight, let's drift off together while exploring over 100 soothing facts about the human body. There's no rush here, no test at the end. Just soft words and quiet wonders to carry you into rest. Welcome to Drowsy Discovery. I am glad you found your way here tonight. Perhaps you have had a long day. Perhaps you simply enjoy falling asleep to thoughts about the remarkable machine that carries you through life. Either way, this space is for you. There is something profoundly peaceful about contemplating the inner world. Every fact about the body is a reminder that you carry within you the most complex structure in the known universe. And it is always working, always dreaming, always creating the world you experience. You are part of this story. The oxygen in your lungs has passed through countless leaves. The iron in your blood was forged in ancient stars. The atoms that make your cells have been part of oceans, mountains, and creatures that lived millions of years before you were born. If you enjoy these gentle journeys, I invite you to like, subscribe, or share a thought below. It helps others find their way here too, one sleepy soul at a time. But for now, all you need to do is relax. So settle in. Let your breathing slow. And let the weight of the day drift away like morning mist rising from a quiet lake. The body is patient beyond measure. Cells divide for decades without complaint. Hearts beat for a lifetime without pause. Bones reshape themselves to carry you through every step of your journey. And yet, within all that steadiness, there are sudden bursts of wonder. Nerves that fire faster than Formula One cars. Muscles that vibrate faster than hummingbird wings. Immune cells that remember infections for decades. Tonight, we will wander through this magnificent inner world. We will meet the cells that never sleep and discover processes that shape the very essence of who you are. And we will uncover truths about the human body that may surprise even the most curious minds. But there is no hurry. Your body has been working since before you were born. It can wait a little longer while you rest. Let us begin with the quiet miracle of cellular renewal. Your body creates 25 million new cells every second. The rate of cellular production within your body is so immense that it challenges ordinary comprehension. Every single second, roughly 25 million new cells emerge from division processes occurring simultaneously across your tissues and organs. This constant creation is necessary to replace cells that have reached the end of their functional lifespan or suffered damage during their duties. Different tissues have vastly different turnover rates, with some cells lasting only days while others persist for years or even decades. The intestinal lining replaces itself most rapidly due to the harsh environment it endures, while brain cells and heart muscle cells are replaced far more slowly, if at all. Your bone marrow alone produces roughly 200 billion red blood cells every day, to replace those that have worn out after their 4-month journey through your bloodstream. This ceaseless production continues throughout your life, slowing somewhat with age but never truly stopping. You shed an entire layer of skin every month. The outermost layer of your skin, called the epidermis, is in a constant state of renewal that you rarely notice. Cells born in the deeper layers gradually migrate toward the surface over approximately 2 to 4 weeks, flattening and hardening as they journey upward. By the time these cells reach the surface, they have died and become tough, protective scales called corneocytes that form your visible skin barrier. Each day you shed roughly 30 to 40,000 dead skin cells, which accounts for much of the dust found in homes and buildings. Over the course of a month, your entire outer skin layer has been replaced by fresh cells from below. This continuous renewal helps repair minor damage from sunlight, friction, and environmental stress. The process also helps eliminate bacteria and other microorganisms that have attached to the dying cells, providing a form of passive immune defense. Your stomach regenerates its lining every 3 to 4 days. The interior of your stomach is lined with a layer of cells that faces one of the harshest environments in your body. Hydrochloric acid, strong enough to dissolve metal, bathes these cells continuously as they work to break down the food you eat. To survive this chemical assault, the stomach lining replaces itself more rapidly than almost any other tissue in your body. Specialized stem cells at the base of gastric glands constantly divide and push new cells upward to replace those that have been damaged by acid and digestive enzymes. This rapid turnover means that the cells you have today are entirely different from those that lined your stomach just a few days ago. A thick layer of mucus protects the underlying cells while this replacement occurs, creating a barrier between living tissue and digestive juices. When this protective system fails, ulcers can develop where acid reaches and damages the vulnerable tissue beneath. You replace your entire skeleton every 10 years. Despite their appearance of permanence, your bones are dynamic living tissues that constantly break themselves down and rebuild. Specialized cells called osteoclasts dissolve old bone tissue, while osteoblasts lay down fresh bone matrix in a carefully coordinated process called remodeling. This continuous renewal allows your skeleton to repair microscopic damage from daily stress and adapt its structure to the loads you place upon it. Over approximately 10 years, every atom of calcium and phosphorus in your bones has been replaced with new material absorbed from your diet. The rate of bone turnover varies throughout life, proceeding most rapidly during childhood and slowing considerably in old age. Regular physical activity stimulates bone formation because the mechanical stress signals osteoblasts to strengthen areas under load. This remarkable plasticity means that the skeleton supporting you today is literally not the same one you had a decade ago. Your liver can regrow from just a quarter of its tissue. The liver possesses a remarkable regenerative capacity, unmatched by any other solid organ in the human body. If up to 75% of the liver is removed or destroyed, the remaining tissue can regrow to restore the organ to its original size within weeks. This extraordinary ability allows living donor liver transplants, where a portion of a healthy person's liver is given to a patient in need, with both donor and recipient eventually having full-sized livers. The regeneration does not recreate the original anatomical lobes, but rather expands the remaining tissue to restore functional mass. This capacity evolved because the liver performs so many essential functions that survival without it is impossible. Hepatocytes, the main liver cells, normally divide rarely but can be triggered to proliferate rapidly when the organ suffers damage. Ancient cultures seem to sense this regenerative power, as reflected in the myth of Prometheus, whose liver regrew each night after an eagle consumed it during the day. You generate around 300 billion new cells every day. The total daily production of new cells across all tissues in your body amounts to roughly 300 billion, a number so vast that it equals the number of stars in a large galaxy. Most of these new cells are blood cells produced in your bone marrow, but significant contributions come from your skin, gut lining, and other rapidly renewing tissues. Each new cell must copy approximately 3 billion base pairs of DNA accurately, a process that involves remarkable molecular machinery working at astonishing speed. Despite the astronomical number of cell divisions occurring daily, errors in DNA replication are surprisingly rare, thanks to sophisticated proofreading mechanisms. The cells that make mistakes are usually detected and destroyed before they can cause problems. This massive production continues throughout your life, though the rate declines somewhat with age as stem cell populations shrink. The cells being created right now will become the blood, skin, and gut lining that will serve you in the weeks ahead. Your bones are constantly dissolving and rebuilding. Bone tissue exists in a perpetual state of controlled destruction and reconstruction that maintains skeletal strength throughout your life. Osteoclasts are specialized cells that secrete acid and enzymes to dissolve small areas of bone, creating microscopic cavities in the tissue. Following close behind, osteoblasts fill these cavities with fresh bone matrix that eventually hardens with calcium and phosphorus. This process of remodeling serves multiple purposes beyond simple maintenance, allowing bones to adapt their architecture to the mechanical stresses they experience. Areas under repeated load become denser and stronger, while regions that bear little stress may become thinner over time. The remodeling cycle also releases calcium into your bloodstream when supplies run low, making bones a mineral reservoir for the entire body. When the balance tips toward too much destruction or too little formation, conditions like osteoporosis can develop, leaving bones fragile and prone to fracture. Bones are ounce for ounce stronger than steel. When comparing materials of equal weight, bone proves stronger than mild steel in its ability to resist breaking under load. This remarkable strength comes from bone's composite structure, which combines a hard mineral matrix with flexible protein fibers in a way that outperforms either material alone. The mineral component, primarily calcium phosphate crystals, provides rigidity and resistance to compression, while collagen fibers provide tensile strength and flexibility. This combination allows bone to absorb energy from impacts that would shatter a purely mineral structure, and to resist bending forces that would deform pure metal. Engineers have studied bone architecture for inspiration in designing lightweight yet strong materials for aircraft and buildings. The hollow structure of long bones further increases their strength-to-weight ratio, providing maximum resistance to bending with minimum material. Nature has spent hundreds of millions of years refining this design to create a skeletal material that is both light enough to move and tough enough to last a lifetime. Your bones are hollow, but incredibly strong. Long bones like those in your arms and legs are not solid cylinders, but hollow tubes filled with marrow, a design that maximizes strength while minimizing weight. Engineers recognize this hollow tube structure as one of the most efficient ways to resist bending, providing far more rigidity per unit of weight than a solid rod would offer. The outer layer of compact bone provides the primary structural strength, while the central cavity houses bone marrow where blood cells are produced. This arrangement means your skeleton can support your weight and withstand daily impacts while remaining light enough for efficient movement. The walls of long bones are thickest where bending stresses are greatest, and thinner where less strength is needed. If human bones were solid, your skeleton would weigh significantly more and require substantially more energy to move. This elegant hollow design also leaves room for the blood cell factories that keep you alive. Bones stop growing in length but not in width. Once you reach adulthood, the growth plates near the ends of your long bones close permanently, ending any further increase in height. These cartilaginous zones where new bone length is added eventually become bone themselves, sealing off the mechanism that made you taller during childhood and adolescence. However, bones retain the ability to increase in width throughout life through a process called appositional growth. New bone can be laid down on the outer surface by cells in the membrane covering the bone, while the inner cavity may expand through resorption. This capacity allows bones to thicken and strengthen in response to increased mechanical loading, which is why regular exercise can increase bone density even in older adults. Athletes and manual laborers often develop thicker bones in the limbs they use most heavily. The ongoing potential for width changes means your skeleton continues adapting to your lifestyle long after you stop growing taller. Your bones contain living tissue and stem cells. Far from being inert mineral structures, bones are dynamic organs filled with living cells, blood vessels, and nerves. The hard exterior conceals a bustling community of osteocytes embedded in the mineral matrix, connected to each other through microscopic channels that allow communication and nutrient exchange. Bone marrow occupying the interior cavities contains stem cells capable of generating all the blood cells circulating through your body. These same stem cells have the potential to develop into bone, cartilage, and fat cells under appropriate conditions, a versatility that makes them valuable in medical treatments. Blood vessels penetrating the bone bring oxygen and nutrients to the living cells within, while removing waste products. Nerves running through bone register pain when fractures occur or inflammation develops. The living nature of bone tissue explains why broken bones can heal and why bones can weaken when blood supply is interrupted. Human bones are stronger under compression than under tension. Bone is remarkable for its ability to resist compression, the force that pushes material together, far better than it resists tension, the force that pulls material apart. This property reflects the nature of the mineral matrix, which handles crushing forces excellently, but can crack when stretched. The architecture of your skeleton takes advantage of this asymmetry, positioning bones so that they primarily experience compression during normal activities, like standing and walking. The gentle curves of your spine and the angles of your hip joints direct forces so that compressive loads predominate along bone shafts. When bones do experience significant tension, the collagen fibers embedded in the matrix help resist stretching, but fractures still occur more easily under tensile or bending loads. Athletes who subject their bones to unusual forces sometimes suffer stress fractures, where repeated tension has weakened the tissue. Understanding these mechanical properties helps surgeons design implants and rehabilitation programs. Your heart beats around 100,000 times per day. The ceaseless rhythm of your heart accumulates to roughly 100,000 contractions every day, a total that amounts to more than 2.5 billion beats over an average lifetime. Each beat propels approximately 70 milliliters of blood into your circulatory system, adding up to over 7,000 liters pumped every day. This continuous work happens without conscious effort, controlled by electrical impulses that originate within the heart itself, rather than from the brain. The muscle fibers of your heart are unique in their ability to contract rhythmically, without ever truly resting, a feat no other muscle in your body can match. Between each beat, the heart relaxes just long enough to refill with blood before squeezing again. The entire cycle from one contraction to the next takes less than a second, yet the precision of this timing keeps blood flowing smoothly through your vessels. This tireless beating begins before birth and continues without a single break until the moment of death. The heart creates enough pressure to squirt blood 30 feet. When the left ventricle contracts, it generates enough pressure to propel blood from an artery a distance of approximately 10 meters if the vessel were open to the air. This impressive force is necessary to push blood through the vast network of arteries, capillaries, and veins that serve every tissue in your body. The pressure must remain high enough to overcome the resistance of narrow blood vessels while maintaining steady flow to distant regions like your toes. Blood pressure measured at your arm reflects this forceful output, with the higher number representing the peak pressure during contraction and the lower number showing the pressure between beats. The thick muscular wall of the left ventricle is specifically designed to generate this high-pressure output, while the right ventricle produces lower pressure suitable for the shorter journey to the lungs. This hydraulic power operates continuously, driving blood through roughly 100,000 kilometers of vessels. The heart has its own electrical system and can beat outside the body. Unlike most muscles that require signals from the nervous system to contract, the heart contains specialized pacemaker cells that generate their own electrical impulses. This intrinsic electrical system means the heart can continue beating even when completely disconnected from the brain and body, a property essential for heart transplantation. The sinoatrial node in the right atrium initiates each heartbeat, sending electrical waves across the heart muscle that coordinate the precise timing of contraction. These impulses travel through specialized conducting pathways that ensure the atria contract before the ventricles, optimizing blood flow through the chambers. A heart removed for transplant can continue beating for hours if kept in appropriate conditions with adequate oxygen and nutrients. This electrical independence evolved because the heart must beat continuously without waiting for nerve signals that might be delayed in stressful situations. The heart's autonomous rhythm ensures that circulation continues even when other body systems are compromised. Your body has a backup system for controlling your heartbeat. The heart contains multiple redundant pacemaker sites that can take over if the primary pacemaker fails, providing a safety net for this critical organ. The sinoatrial node normally controls heart rhythm, but if it stops functioning, the atrioventricular node can assume command at a somewhat slower rate. Even if both nodes fail, individual heart muscle fibers can generate their own slower rhythm to maintain at least basic circulation. This redundancy reflects the critical importance of maintaining blood flow, as even brief interruptions can cause brain damage or death. Modern pacemaker devices implanted in patients with heart rhythm disorders mimic this natural backup system by monitoring the heart and providing electrical stimulation when needed. The nervous system also provides external input to speed up or slow down the heart in response to exercise, stress, or rest, but these signals modify rather than initiate the heartbeat. This layered control system keeps your heart beating reliably throughout decades of continuous operation. Every heartbeat triggers a pressure wave that reaches your brain. Each contraction of your heart sends a pulse of pressure through your arteries that reaches your brain in a fraction of a second. This pressure wave causes slight mechanical deformation of blood vessels and surrounding tissues throughout your body, including structures within your skull. Research suggests that the brain does not simply ignore these rhythmic pulses, but actually synchronizes some of its activity with the cardiac cycle. Certain cognitive processes and emotional responses appear to vary slightly, depending on the phase of the heartbeat at which they occur. The baroreceptors in your arteries detect each pressure wave and send signals to your brain that help regulate blood pressure and heart rate. This continuous feedback loop between heart and brain means the two organs are in constant communication, each influencing the other. The ancient intuition that emotions are felt in the heart may reflect a genuine physiological connection between cardiac rhythm and conscious experience. Some blood vessels are so small that red blood cells must squeeze through one at a time. Capillaries, the smallest blood vessels in your body, have internal diameters smaller than the width of a single red blood cell. This tight fit forces red blood cells to deform as they squeeze through, folding their disc-shaped bodies to pass single file through these microscopic channels. The intimate contact between each blood cell and the capillary wall maximizes the exchange of oxygen and nutrients with surrounding tissues. A red blood cell may take less than a second to traverse a typical capillary, but in that brief moment it releases oxygen and absorbs carbon dioxide from adjacent cells. The total length of capillaries in your body is estimated at roughly 100,000 kilometers, providing an enormous surface area for this vital exchange. If all the capillaries in your body were laid end to end, they would circle the Earth more than twice. This vast microscopic network reaches within a few cell widths of nearly every living cell in your body. Red blood cells carry oxygen but have no nucleus. Mature human red blood cells are among the only cells in your body that lack a nucleus, having expelled this genetic command center during their development. This unusual sacrifice creates more internal space for hemoglobin, the iron-containing protein that binds and carries oxygen from your lungs to every tissue. Each red blood cell contains roughly 270 million hemoglobin molecules, giving these tiny cells an enormous oxygen-carrying capacity. The absence of a nucleus also gives red blood cells their distinctive flexible disc shape, which allows them to squeeze through the narrowest capillaries. Without a nucleus, these cells cannot divide or repair themselves, so they have a limited lifespan of approximately 120 days. Your spleen filters out and destroys aging red blood cells that have become too stiff to navigate the circulatory system. The iron from broken down cells is recycled and sent to the bone marrow to equip the next generation of oxygen carriers. Human blood comes in many rare types beyond AB, AB and O. While most people know about the ABO blood group system, human blood actually contains dozens of different antigen systems that determine compatibility for transfusion. Scientists have identified more than 30 blood group systems and over 300 individual antigens that can appear on the surface of red blood cells. Some rare blood types are found in only a handful of people worldwide, making finding compatible donors extremely challenging. The Rh system, which classifies blood as positive or negative, is the second most important system after ABO for transfusion purposes. Beyond these major systems, antigens like the KEL, Duffy and KID groups can cause serious reactions if mismatched blood is transfused. Some people carry such rare combinations of blood antigens that they can only receive blood from a few other individuals on Earth. Blood banks around the world maintain frozen reserves of rare blood types to ensure these unusual patients can receive safe transfusions when needed. Your immune system remembers past infections for decades. After your body successfully fights off an infection, specialized memory cells retain information about the invader for years, or even a lifetime. These memory B cells and memory T cells persist in your lymph nodes and bloodstream, ready to mount a rapid response if the same pathogen appears again. When reinfection occurs, this immunological memory allows your body to produce antibodies and deploy defensive cells within hours rather than the days or weeks required for a first encounter. This principle underlies vaccination, which trains your immune system to recognize pathogens without causing actual disease. Some vaccines provide protection lasting decades, while others require boosters because the memory cells eventually decline in number. The remarkable longevity of immune memory explains why you usually only get diseases like measles or chickenpox once in your lifetime. Your immune system maintains a library of past battles, ready to deploy proven defenses against enemies it has faced before. White blood cells have a built-in navigation system to find infections. When bacteria invade your body, damaged cells and immune signals create chemical trails that white blood cells follow to locate the site of infection. This process, called chemotaxis, allows immune cells to navigate through tissues and blood vessels towards sources of inflammation with remarkable precision. White blood cells can detect extremely low concentrations of these chemical signals and move along the gradient toward higher concentrations. The cells change their shape dynamically as they migrate, extending projections in the direction of the chemical source and pulling themselves forward. Some white blood cells can even squeeze between the cells lining blood vessel walls to enter tissues where infection is occurring. This sophisticated navigation system ensures that defensive cells concentrate exactly where they are needed rather than wandering randomly. Without this targeting mechanism, your immune system would be far less effective at containing infections before they spread. Bone marrow is the source of all red blood cells. The soft, spongy tissue filling the interior cavities of your bones is responsible for producing virtually all the blood cells circulating through your body. Red bone marrow contains hematopoietic stem cells that can develop into any type of blood cell, including red cells, white cells, and platelets. These stem cells divide continuously throughout your life, replacing the billions of blood cells that are destroyed or worn out each day. In infants, red marrow fills nearly all bones, but by adulthood it has retreated to the pelvis, spine, ribs, sternum, and ends of major long bones. The remaining bone cavities contain yellow marrow, which stores fat but can convert back to red marrow if the body needs to increase blood cell production. Bone marrow transplants can save the lives of patients whose own marrow has failed or been damaged by cancer treatment. The marrow produces roughly 500 billion blood cells every day, making it one of the most productive tissues in your body. The brain produces enough electricity to power a small light bulb. The electrical activity generated by the collective firing of billions of neurons in your brain produces roughly 12 to 25 watts of power, sufficient to illuminate a dim light bulb. This seemingly modest output belies the incredible computational work being performed, as your brain processes more information than any computer ever built, while consuming just a fraction of the energy. The brain's electrical activity can be measured through electroencephalography, which detects the combined signals from millions of neurons firing in rhythmic patterns. These brainwaves vary with mental states, showing different frequencies during sleep, relaxation, concentration, and intense thought. The efficiency with which your brain accomplishes complex tasks, using only the power of a small bulb, far exceeds that of artificial computers performing similar functions. Modern computer chips performing equivalent calculations would require thousands of watts and elaborate cooling systems. Your brain achieves its remarkable feats while generating barely enough heat to warm your skull slightly. Your brain has more than 100 billion neurons. The human brain contains approximately 86 billion neurons, a number so vast that counting them one per second would take nearly 3,000 years. Each of these neurons can connect with thousands of others, creating an interconnected network of almost incomprehensible complexity. Neurons communicate through a combination of electrical impulses and chemical signals, passing information across tiny gaps called synapses. Different regions of the brain contain specialized populations of neurons responsible for distinct functions. from processing vision to coordinating movement to generating language. Beyond the neurons, your brain contains roughly equal numbers of glial cells that support, nourish, and protect the neural network. The sheer density of cells within your skull is remarkable, with each cubic millimeter of brain tissue containing roughly 50,000 neurons and hundreds of thousands of synapses. This biological computer, weighing only about 1.4 kilograms, generates every thought, memory, and experience you have ever had. There are more connections in your brain than stars in the Milky Way. The synaptic connections between neurons in your brain number in the hundreds of trillions, vastly exceeding the estimated 200 billion stars in our galaxy. Each of your 86 billion neurons forms an average of 7,000 synaptic connections with other neurons, creating a web of staggering complexity. These connections are not fixed but constantly changing as you learn and experience life, with new synapses forming and unused ones being pruned away. The total number of possible neural connection patterns exceeds the number of atoms in the known universe, giving the brain almost unlimited potential for storing information and generating unique thoughts. This synaptic network is the physical substrate of your personality, memories, skills, and every aspect of who you are. The connections strengthen when repeatedly activated, forming the basis of learning and habit formation. Every experience you have subtly reshapes this cosmic web of connections inside your skull. Nerve impulses travel faster than a Formula One car. The fastest nerve fibers in your body conduct electrical impulses at speeds exceeding 100 meters per second, outpacing a Formula One car at full throttle. These rapid signals travel along myelinated axons, nerve fibers wrapped in an insulating fatty sheath that allows impulses to jump between gaps rather than flowing continuously. The speed of nerve conduction varies considerably depending on the type of fiber, with some slow pain signals traveling at less than 1 meter per second. Fast conduction evolved where rapid response is essential, such as in the motor nerves controlling movement and the sensory nerves that detect sudden touches or sounds. The delay between sensing a stimulus and becoming aware of it is typically just a few milliseconds, though you rarely notice this brief lag. Reflex arcs that bypass the brain can respond even faster, producing protective movements before conscious awareness catches up. This remarkable signaling speed allows your nervous system to coordinate complex movements with split-second precision. The brain uses more energy than any other organ. Despite accounting for only about 2% of body weight, your brain consumes roughly 20% of the body's total energy output. This disproportionate energy demand reflects the tremendous metabolic activity required to maintain neural function and communication. Neurons require constant supplies of oxygen and glucose to power the ion pumps that maintain the electrical gradients essential for signaling. When blood flow to the brain is interrupted for even a few seconds, consciousness begins to fade, and permanent damage can occur within minutes. Brain imaging techniques like PET and fMRI detect changes in blood flow and metabolism that reveal which regions are most active during different tasks. The brain's energy consumption remains remarkably constant, whether you are thinking intensely or at rest, as most neural activity is devoted to maintaining the complex network rather than processing specific thoughts. This hungry organ evolved its extraordinary demands because the survival advantages of intelligence justified the metabolic cost. Your brain does not feel pain because it has no pain receptors. The brain tissue itself contains no nociceptors, the sensory receptors that detect potentially harmful stimuli and generate the sensation of pain. This seemingly paradoxical design means that surgeons can operate on the brain of a conscious patient without causing pain to the neural tissue itself. Pain perceived during headaches originates from the blood vessels, membranes, and muscles surrounding the brain, rather than from the brain tissue. The absence of pain receptors in the brain makes evolutionary sense, because the organ is protected inside the skull, and would have no opportunity to take protective action if it could feel damage. Local anesthesia or sedation is still needed during brain surgery to numb the scalp and skull during the opening procedure. Once the brain is exposed, surgeons can probe and manipulate tissue while the patient remains awake and answers questions to help map critical regions. This remarkable insensitivity allows doctors to treat conditions that would otherwise be inaccessible. The brain can rewire itself in response to damage. When injury or disease destroys neurons in one part of the brain, other regions can sometimes take over the lost functions through a process called neuroplasticity. This remarkable adaptability allows stroke survivors to regain movement and speech as undamaged areas gradually assume the roles of destroyed tissue. The rewiring process involves the strengthening of existing connections and the formation of entirely new neural pathways that bypass the damaged region. Recovery is most dramatic when rehabilitation begins quickly after injury, taking advantage of the brain's heightened plasticity during the early healing period. Children show the most impressive recovery from brain damage because their developing brains retain more potential for reorganization than adult brains. Even in adulthood, however, dedicated practice and therapy can drive meaningful structural changes in the brain. This capacity for self-repair gives hope to patients recovering from injuries that once would have caused permanent disability. You can function with half a brain and sometimes better than before. In cases of severe epilepsy that cannot be controlled with medication, surgeons sometimes remove an entire hemisphere of the brain in a procedure called hemispherectomy. Despite losing half their brain tissue, many patients not only survive but thrive, with some experiencing improvements in cognitive function as seizures cease. Children who undergo this radical surgery can develop remarkably normal language and motor skills, as the remaining hemisphere assumes functions normally handled by both sides. The intact hemisphere rewires extensively, compensating for the lost tissue through enhanced plasticity that would not occur in an older brain. Some patients show better scores on cognitive tests after surgery because the constant electrical chaos of seizures had been disrupting all brain function. Adults can also survive hemispherectomy, though their recovery is typically less complete than that seen in children. These extraordinary cases demonstrate that the brain possesses far more redundancy and adaptability than was once believed possible. The brain can generate hallucinations with no external input. Your brain creates sensory experiences entirely from internal activity, generating sights, sounds, and sensations in the complete absence of external stimuli. This capacity becomes dramatically apparent during dreams, when vivid visual scenes unfold while your eyes are closed, and the visual cortex receives no input from the outside world. Hallucinations occurring in waking life involve similar activation of sensory processing regions, without corresponding external signals. People who lose their sight sometimes develop Charles Bonnet syndrome, experiencing detailed visual hallucinations as the visual cortex, deprived of input, generates its own activity. Sensory deprivation experiments reveal that the brain will manufacture perceptions when cut off from external stimulation for extended periods. These phenomena suggest that normal perception involves the brain continuously generating models of reality that are updated rather than created by sensory input. Your experience of the world is fundamentally a construction of your brain, informed but not directly caused by signals from your senses. The brain can simulate sensory experience purely through thought. Mental imagery activates many of the same brain regions that respond to actual sensory input, allowing you to see, hear, and feel things that exist only in your imagination. When you visualize a familiar face, your visual cortex shows activity patterns similar to those produced when actually viewing that face. This overlap between imagination and perception explains why mental rehearsal can improve physical skills, as the brain regions controlling movement activate even when you only imagine performing an action. Athletes and musicians use mental practice to reinforce motor patterns without physically executing movements. The brain areas for imagined and perceived experiences are so closely linked that vivid imagination can sometimes be confused with actual memory. This remarkable capacity for internal simulation allows you to plan future actions, relive past experiences, and create entirely fictional scenarios within your mind. The boundary between imagined and perceived reality is far less distinct than most people realize. Memory is not stored in a single place, but distributed across networks. Contrary to early theories that memories might be stored in specific locations like files in a cabinet, research has revealed that memories are distributed across widespread neural networks. Different aspects of a single memory are encoded in different brain regions, with visual elements stored in visual cortex, sounds in auditory cortex, emotions in the limbic system, and spatial context in the hippocampus. Recalling a memory involves the coordinated reactivation of all these dispersed components, reassembling the experience from scattered fragments. This distributed storage makes memories remarkably resilient, as damage to one region may degrade but not entirely destroy a memory. However, it also makes memories prone to modification during recall, as the reassembly process can introduce new elements or alter existing ones. Each time you remember an event, you may subtly change the memory trace, explaining why recollections can shift over time. Your memories are not recordings but reconstructions, rebuilt each time from distributed pieces. Your brain is sometimes more active while you sleep than when awake. During certain stages of sleep, particularly rapid eye movement sleep, your brain shows activity levels matching or exceeding those observed during waking consciousness. This nocturnal activity is not random noise, but serves essential functions, including memory consolidation, emotional processing, and neural maintenance. The sleeping brain replays and reorganizes information acquired during the day, strengthening important memories while clearing out unnecessary data. Some neural regions that remain relatively quiet during focused waking activity become intensely active during sleep, handling housekeeping tasks that cannot be performed while the brain is busy with conscious thought. The glymphatic system, which clears metabolic waste from the brain, operates most efficiently during deep sleep, when the spaces between cells expand to allow better fluid flow. Sleep deprivation profoundly impairs cognitive function precisely because the brain needs this active resting state to maintain itself. The busy activity of your sleeping brain explains why adequate quality sleep is essential for mental health and performance. Dreams can encode memories, solve problems, and rehearse fears. The content of dreams appears to serve multiple important functions beyond mere entertainment during sleep. Research suggests that dreaming helps consolidate memories by replaying and integrating recent experiences with older knowledge stored in long-term memory. Many famous scientific discoveries and creative breakthroughs have come during or immediately after sleep, suggesting that dreams can work on problems that stump the waking mind. The bizarre scenarios common in dreams may serve as a form of threat simulation, allowing you to rehearse responses to dangers in a safe environment. Dreams involving stressful situations may help regulate emotions by allowing the brain to process difficult experiences with reduced activity in regions that control anxiety. The prefrontal cortex, which oversees rational thought and self-awareness, shows reduced activity during dreaming, perhaps explaining the strange logic that prevails in dream worlds. Whatever their full purpose, dreams are clearly not meaningless, but rather the visible traces of essential cognitive work. You grow taller in the morning than in the evening. Over the course of each day, your height decreases by roughly 1 to 2 centimeters, as the cartilage discs between your vertebrae compress under the weight of your body. These intervertebral discs contain a gel-like core that absorbs water during the night when you lie down, and pressure is relieved. The rehydration causes the discs to expand, restoring the height lost during the previous day and making you measurably taller when you first wake up. Astronauts returning from extended space missions can be several centimeters taller than when they left, because the lack of gravity allows their spinal discs to expand without the usual daily compression. The daily height fluctuation explains why measurements taken at different times of day can give slightly different results. As you age, the discs lose some of their ability to rehydrate fully, contributing to the gradual height loss that occurs over decades. This rhythmic compression and expansion is one of many ways your body changes throughout the day. The body has a circadian clock in almost every organ. Beyond the master circadian clock in your brain, nearly every organ and tissue in your body contains its own local timekeeping mechanism. These peripheral clocks synchronize with the central clock, but can operate semi-independently, coordinating local functions with the broader rhythms of the body. Your liver has a clock that regulates metabolism to anticipate mealtimes, while your heart has a clock that adjusts cardiac output for the demands of day versus night. The gut, kidneys, skin, and muscles all contain circadian oscillators that time their activities to optimal periods within the daily cycle. When these local clocks fall out of sync with each other, or with the external day-night cycle, health problems can result. Shift workers and frequent travelers often experience the consequences of misaligned circadian rhythms, including metabolic disturbances and increased disease risk. The distributed nature of your body's timekeeping reveals how deeply the rhythm of the day is embedded in your physiology. Your body temperature varies by time of day and activity. Normal body temperature is not a fixed value but fluctuates rhythmically throughout the day by nearly 1 degree Celsius. Core temperature typically reaches its lowest point in the early morning hours, when you are deeply asleep, and peaks in the late afternoon or early evening. This temperature cycle is controlled by your circadian clock and influences alertness, reaction time, and physical performance throughout the day. The evening temperature increase is partially why many people feel most energetic in the afternoon and early evening. Exercise can temporarily raise body temperature significantly, while illness can override the normal daily rhythm with fever. The drop in core temperature that occurs before sleep helps trigger drowsiness, and is one reason why a cool bedroom promotes better rest. Your internal thermostat constantly adjusts blood flow to the skin and metabolic heat production to maintain core temperature within a narrow range, despite these planned fluctuations. Your eyes can distinguish over 10 million colors. The human visual system can differentiate between approximately 10 million distinct colors, a remarkable perceptual capacity generated by just 3 types of color-sensitive cone cells in your retina. Each cone type responds most strongly to either red, green, or blue wavelengths of light, and the brain calculates perceived color from the combined signals of all 3. The millions of distinguishable hues arise from the countless possible combinations of stimulation levels across the 3 cone populations. Some individuals have 4 types of cone cells, potentially allowing them to perceive colors invisible to normal eyes, though most people possess the standard 3. The colors you perceive are not properties of light itself, but constructions of your visual cortex interpreting wavelength information. Animals with different numbers or types of photoreceptors experience colors that humans cannot imagine. This extraordinary discriminating ability evolved to help our ancestors identify ripe fruit, detect predators, and navigate complex natural environments. The cornea has no blood supply and gets oxygen from the air. The transparent cornea covering the front of your eye must remain perfectly clear to focus light, so it cannot contain the blood vessels that supply most other tissues with oxygen. Instead, the cornea absorbs oxygen directly from the air across its outer surface, one of the few tissues in your body capable of this atmospheric exchange. The inner layers of the cornea receive some nutrients and oxygen from the aqueous humor, the clear fluid that fills the chamber behind it. This unique arrangement makes the cornea vulnerable to oxygen deprivation when covered by contact lenses, which is why wearing contacts too long can cause discomfort and damage. Modern contact lens materials are designed to allow more oxygen transmission to reduce the risk of corneal problems. The absence of blood vessels also means the cornea heals differently from other tissues, with scarring that can permanently cloud vision. The transparency that gives your cornea its essential function comes at the cost of a precarious metabolic supply. Your eyes are never the same size throughout life. Your eyeballs grow significantly during childhood, beginning at about 16 millimeters in diameter at birth, and reaching their adult size of roughly 24 millimeters by adolescence. This growth must be precisely controlled, because even small changes in eye length affect how clearly images focus on the retina. If the eye grows too long, the result is myopia or nearsightedness, as images focus in front of the retina rather than directly on it. The rapid increase in childhood myopia worldwide may be partly related to environmental factors that disrupt normal eye development. After reaching adult size, the eyes remain relatively stable, though changes in the lens and other structures continue throughout life. The length of your eye at any age reflects the interplay of genetic programming and environmental influences during development. Understanding how the eye controls its own growth could lead to new treatments for preventing vision problems before they develop. You have cells in your eye that detect only horizontal or vertical lines. The visual cortex contains neurons that respond specifically to edges oriented in particular directions, with some cells firing only for horizontal lines, others only for vertical, and still others for various diagonal angles. This orientation selectivity begins with the way retinal signals are combined in the brain, creating detectors for specific geometric features of the visual scene. Nobel Prize-winning research by David Hubel and Torsten Wiesel revealed this remarkable specialization through studies where they recorded from individual neurons, while presenting different visual stimuli. The brain builds complex visual perception from simple feature detectors that respond to edges, angles, motion, and color, combining their outputs to recognize objects and scenes. Animals raised without exposure to certain orientations during critical periods of development fail to develop the corresponding orientation-selective cells. This finding demonstrates that visual system wiring depends on visual experience during early life. The cells detecting lines and edges are fundamental building blocks from which all visual recognition is constructed. Some cells in your eye are more sensitive to motion than to light. Your retina contains specialized ganglion cells that respond primarily to movement across the visual field rather than to static images or overall brightness. These motion-detecting cells are particularly sensitive to objects moving in specific directions, firing vigorously when something crosses their receptive field in the preferred direction and barely responding to motion in other directions. This motion sensitivity likely evolved to detect approaching predators or moving prey, situations where immediate awareness of movement is more important than detailed pattern recognition. The signals from motion-sensitive cells travel to brain regions that process movement and guide reflexive eye movements to track moving objects. Your peripheral vision, though poor at resolving detail, remains exquisitely sensitive to motion, which is why you can notice something moving at the edge of your visual field even when you cannot identify what it is. This design allows your eyes to serve as an early warning system for your brain. You blink around 20 times per minute without thinking. The average person blinks roughly 15 to 20 times each minute during waking hours, a rate that amounts to approximately 20,000 blinks per day. Each blink spreads a fresh layer of tears across the cornea, washing away debris and keeping the delicate surface moist and clear. The timing of blinks is not random, but tends to occur at natural breakpoints in attention, such as the end of a sentence while reading or during pauses in conversation. Your blink rate decreases significantly when you focus intently on a task requiring close visual attention, which is why staring at screens can cause dry eyes. Blinking also briefly interrupts visual input, yet you rarely notice these momentary blackouts because your brain suppresses awareness of them. The coordination between blinking and attention reveals that even this seemingly simple reflex is integrated with higher cognitive processes. Most blinks occur automatically without conscious awareness, yet you can also blink voluntarily when desired. Your sense of balance comes from fluid in tiny ear canals. 3 semicircular canals in each inner ear contain fluid that moves whenever your head rotates, providing your brain with precise information about angular motion. The canals are oriented at right angles to each other, allowing them to detect rotation in any direction, whether you nod, shake, or tilt your head. When you turn, the fluid initially lags behind due to inertia, deflecting tiny hair cells that convert the movement into nerve signals. Your brain compares input from the canals in both ears to calculate the exact speed and direction of head rotation. This vestibular system works in concert with vision and sensors in your joints and muscles to maintain balance and spatial orientation. When the signals from these different systems conflict, as can happen on a rocking boat, motion sickness often results. Damage to the semicircular canals can cause severe vertigo as the brain receives incorrect information about movement. The tiny hairs in your ear help detect sound direction. Inside your cochlea, roughly 15,000 microscopic hair cells convert sound vibrations into electrical signals that travel to your brain. These delicate cells are arranged along a spiral membrane that responds to different frequencies along its length, with high-pitched sounds stimulating cells near the base and low-pitched sounds activating cells near the tip. The hair cells themselves contain even tinier projections called stereocilia that bend when sound waves disturb the surrounding fluid. Your brain determines the direction of sounds by comparing timing and intensity differences between signals arriving at each ear. Sounds from the left reach your left ear slightly sooner and louder than your right ear, and your brain calculates the source location from this disparity. The sensitivity of these hairs is remarkable, detecting movements as small as the width of an atom. Unlike many other cells in your body, cochlear hair cells cannot regenerate when damaged, which is why hearing loss from noise exposure or aging is often permanent. The human ear can detect sounds from 20 Hz to 20,000 Hz. Young humans can typically perceive sounds ranging from about 20 vibrations per second at the low end to roughly 20,000 vibrations per second at the high end. This frequency range has evolved to capture the sounds most relevant to human survival and communication, including speech, which falls primarily between 200 and 8,000 Hz. The upper limit of hearing declines with age and noise exposure, with many adults unable to hear frequencies above 15,000 Hz or lower. Children can often hear sounds completely inaudible to their parents, a difference sometimes exploited by devices that emit annoying high-frequency tones intended to deter loitering teenagers. Different animals have vastly different hearing ranges, with elephants detecting infrasound below human perception and bats hearing ultrasound far above it. The range of human hearing represents an evolutionary compromise between the physical constraints of ear structure and the acoustic information most useful to our ancestors. The sounds you hear are just a narrow slice of the vibrations constantly filling the air around you. Some people can hear the movement of their own eyeballs. A rare condition allows certain individuals to perceive an audible sound each time their eyes move, detecting the motion of the eyeball within its socket and the contraction of the muscles controlling eye position. This phenomenon is thought to result from abnormally increased sensitivity of the hearing apparatus or unusual acoustic transmission within the skull. People with this ability describe hearing subtle clicks, whooshes, or other sounds synchronized with each eye movement. For most people, the sounds generated by eye movements are too faint to perceive or are filtered out by the brain as unimportant internal noise. In some cases, this hypersensitivity extends to hearing heartbeat, blood flow, or other internal body sounds that are normally inaudible. The condition can be disturbing when it first develops, but many people adapt to the new perception over time. This unusual ability reveals that many sounds are constantly being generated within our bodies that we normally never detect. You have vestigial muscles that once helped ancestors wiggle their ears. Most humans retain the auricular muscles that allowed our mammalian ancestors to rotate their ears toward sounds, though these muscles have become largely non-functional through disuse over evolutionary time. In many mammals, ear movement is essential for detecting the direction of sounds and expressing emotional states, but humans have largely lost this ability as we came to rely more heavily on rotating our heads. A small percentage of people can still voluntarily wiggle their ears, though the movement serves no practical purpose in modern life. These vestigial muscles respond weakly to electrical stimulation in most people, revealing their dormant presence. The nerves controlling these muscles remain intact, and some researchers have experimented with using ear movement as a hands-free control mechanism for assistive devices. Human ear muscles join other vestigial structures like the appendix and wisdom teeth as evolutionary remnants that hint at our ancestral past. The inability to wiggle your ears is a small reminder of how far humans have diverged from other mammals. Your sense of smell can detect 1 trillion different scents. Recent research suggests that the human olfactory system can distinguish between at least 1 trillion different odors, a staggering number that vastly exceeds earlier estimates of roughly 10,000. This discriminating power emerges from approximately 400 different types of olfactory receptors in your nose, which can be activated in countless combinations by different scent molecules. Each odorant binds to multiple receptor types with varying affinities, and the brain interprets the specific pattern of activation as a particular smell. The olfactory system connects directly to brain regions involved in memory and emotion, explaining why smells can trigger powerful memories more immediately than other senses. Some scents can be detected at concentrations of just a few parts per trillion, revealing extraordinary sensitivity to certain molecules. The wide range of detectable smells evolved partly to help our ancestors identify food, avoid toxins, and navigate social relationships. Your sense of smell is far more sophisticated than the casual sniff might suggest. Taste buds are replaced every 10 to 14 days. The taste receptor cells on your tongue have one of the shortest lifespans of any cells in your body, typically surviving only about 10 to 14 days before being replaced by fresh cells. This rapid turnover means that the taste buds you have today are entirely different from those you had 2 weeks ago. The constant renewal is necessary because taste cells are exposed to the hostile environment of the mouth, where hot food, acidic drinks and mechanical damage from chewing cause regular injury. Each taste bud contains 50 to 100 taste receptor cells that detect the 5 basic tastes: sweet, sour, salty, bitter, and umami. Stem cells at the base of taste buds continually divide to replenish the receptor population as older cells die and are shed. The rapid turnover rate explains why taste can be temporarily altered by illness or certain medications, and then recover fully within weeks. This regenerative capacity ensures that your sense of taste remains sharp throughout life, despite constant wear. You have unique tongue prints just like fingerprints. The surface of your tongue is covered with a pattern of bumps, grooves, and taste receptors that is unique to you, distinguishing your tongue from every other person's on Earth. This uniqueness extends beyond the visible texture to include the geometric shape of the tongue, the distribution of papillae, and the pattern of muscle fibers beneath the surface. Researchers have explored tongue prints as a potential biometric identification method, similar to fingerprints or iris patterns. Unlike fingerprints, the tongue is protected inside the mouth and cannot be easily left behind at crime scenes or forged. The difficulty of making accurate scans has limited practical applications so far, but advances in imaging technology may make tongue-based identification more feasible. Every person's tongue shape reflects the interaction of genetic programming and developmental processes that create slight variations in tissue formation. Your tongue is one more example of how your body is unique in countless subtle ways. Your tears have different chemical compositions, depending on why you cry. The tears streaming down your face during an emotional moment are chemically distinct from the tears that moisten your eyes, or the ones that wash away an irritating speck of dust. Emotional tears contain higher concentrations of stress hormones and protein-based toxins that the body may be releasing during crying. Basal tears that continuously lubricate your eyes contain primarily water, mucus, and antibacterial enzymes that protect against infection. Reflex tears triggered by irritants like onion fumes contain more water and are designed to flush out whatever is bothering the eye. The discovery that emotional tears differ chemically from other types supports the theory that crying serves a detoxification or stress relief function beyond mere eye care. Microscopic examination reveals that different types of tears even crystallize into distinct patterns when dried on slides. The variation in tear chemistry suggests that crying may serve physiological purposes beyond communicating distress to others. You breathe through only one nostril at a time, and it switches. At any moment, one of your nostrils is doing most of the breathing, while the other is relatively congested, and this dominance switches sides every few hours in a process called the nasal cycle. This alternating pattern is controlled by your autonomic nervous system, which cyclically increases blood flow to the erectile tissue inside each nostril, causing temporary swelling that restricts airflow. The cycle means each nostril gets regular rest periods for its mucous membranes to recover and rehydrate. The dominant nostril and the resting one may process air differently, with some research suggesting different effects on brain activity depending on which side is breathing more. Most people remain completely unaware of this cycling unless one nostril becomes permanently blocked by illness or structural abnormality. The cycle continues during sleep, and sleeping position can influence which nostril becomes dominant. This quiet alternation has been occurring throughout your life, without your conscious awareness. You lose up to 1 liter of water every day, just from breathing and skin evaporation. Even without sweating, your body continuously loses water through evaporation from the respiratory tract and skin in a process called insensible water loss. Each breath you exhale carries moisture that was absorbed from the lining of your lungs and airways, amounting to roughly 300 to 400 milliliters per day through breathing alone. Additional water evaporates directly through your skin, even when you are not actively sweating, adding several hundred more milliliters to the daily total. This constant water loss explains why you feel thirsty, even on cool days when you have not exerted yourself. In dry environments or at high altitude, insensible losses can increase substantially, as drier air and faster breathing pull more moisture from your body. The loss from breathing is visible on cold days when your warm, moist exhaled air condenses into a visible cloud. Replacing these invisible losses is essential for maintaining proper hydration and body function. You produce about 1 liter of mucus per day. Your respiratory system produces approximately 1 liter of mucus every day, a substance that plays essential protective roles despite its unappealing reputation. Mucus lines your airways from nose to bronchi, trapping dust, pollen, bacteria, and viruses before they can reach your delicate lung tissue. Tiny hair-like cilia on the cells lining your airways beat rhythmically to move this mucus upward toward your throat, where it is unconsciously swallowed and destroyed by stomach acid. This mucociliary escalator operates continuously, clearing debris from your lungs without you ever noticing. During respiratory infections, mucus production increases dramatically and its consistency changes, becoming thicker as immune cells and antibodies flood into the secretions. The color of mucus can indicate the nature of an infection, though the relationship is less reliable than commonly believed. This continuous river of protective slime is one of your body's first lines of defense against airborne threats. Human vocal cords vibrate faster than a hummingbird's wings. When you speak or sing, your vocal cords vibrate at frequencies ranging from roughly 80 vibrations per second for a deep male voice, to over 1,000 vibrations per second for the highest notes of a soprano. The fastest hummingbird wing beats reach only about 80 beats per second during hovering flight, making your vocal cords capable of far more rapid oscillation. These vibrations are produced by air from your lungs passing between the closed vocal cords, causing them to flutter open and closed at precisely controlled frequencies. The pitch of your voice is determined by the tension and length of your vocal cords, which you adjust using tiny muscles in your larynx. The fundamental frequency produced by the cords is modified by the shape of your throat, mouth, and nasal passages to create the full range of human speech sounds. Trained singers can control their vocal cord tension with extraordinary precision, producing pitches accurate to within a fraction of a semitone. This remarkable vibratory system allows humans to produce the complex sounds that make language and music possible. You have more bacteria in your gut than human cells in your body. Your gastrointestinal tract houses trillions of microorganisms, primarily bacteria, that collectively form a complex ecosystem known as the gut microbiome. Recent estimates suggest the ratio of bacterial to human cells is close to 1:1, though earlier estimates placed bacteria at an even higher proportion. Despite their microscopic size, these microbes collectively weigh between 1 and 2 kilograms and contain millions of genes compared to the roughly 20,000 in the human genome. These bacterial inhabitants are not mere passengers but essential partners that digest certain foods you cannot process, produce vitamins, train your immune system, and synthesize signaling molecules that affect mood and behavior. The composition of your gut microbiome is unique to you, shaped by your birth method, diet, environment, and health history. Disruptions to this microbial community through antibiotics, illness, or poor diet can have cascading effects on digestion, immunity, and even mental health. Your body and its bacterial partners have evolved together in a relationship of mutual dependence. Your intestines are over 20 feet long. Your small and large intestines together form a remarkable length of digestive tubing that measures approximately 20 to 25 feet, or roughly 6 to 7 meters, when stretched out after death. The small intestine accounts for most of this length at around 20 feet, while the large intestine adds another 5 feet to the total. This extraordinary length is necessary because most of your nutrient absorption occurs in the small intestine, and more surface area means more opportunity to extract nutrition from food. The interior surface is further amplified by circular folds, finger-like projections called villi, and microscopic projections on each villus called microvilli, together increasing the absorptive surface to roughly 250 square meters. Food typically takes 3 to 5 hours to transit the small intestine, during which enzymes break down nutrients and the intestinal lining absorbs them into the bloodstream. The intestine manages to fit this enormous length inside your abdomen through tight coiling and folding. This architectural marvel of folding and surface amplification ensures you extract maximum nutrition from every meal. The acid in your stomach can dissolve razor blades. Hydrochloric acid produced by the cells lining your stomach creates an extremely acidic environment with a pH between 1 and 3, acidic enough to dissolve thin metal objects including razor blades over time. This powerful acid serves multiple essential functions, including killing harmful bacteria and pathogens that enter with food, activating digestive enzymes, and beginning the breakdown of proteins. Your stomach protects itself from this caustic environment by producing a thick layer of alkaline mucus that forms a barrier between the acid and the stomach wall. When this protective system fails, the acid can damage the stomach lining, leading to ulcers and painful digestive conditions. The cells producing stomach acid must be constantly renewed, with the entire stomach lining replacing itself roughly every 3 to 4 days. The strength of stomach acid is remarkable, considering the delicate tissues of your body. It contacts only millimeters away from unprotected organs. This carefully controlled balance between destructive acid and protective mucus is essential for both digestion and survival. You produce enough saliva each year to fill 2 bathtubs. Your salivary glands produce between 1 and 2 liters of saliva every day, adding up to roughly 500 to 700 liters per year, enough to fill a couple of standard bathtubs. Saliva is far more than simple water, containing enzymes that begin the digestion of starches while food is still in your mouth, breaking down complex carbohydrates into simpler sugars. It also contains antibacterial compounds that help protect your teeth and gums from infection, and proteins that buffer acids to prevent tooth decay. The lubrication provided by saliva is essential for comfortable chewing and swallowing, which explains why dry mouth makes eating difficult and unpleasant. Saliva production is controlled by your autonomic nervous system, increasing when you smell, see, or think about appetizing food, and decreasing during stress or dehydration. The famous Pavlovian conditioning experiments demonstrated how salivation could be triggered by learned associations, rather than the presence of actual food. This humble fluid performs dozens of essential functions that most people never consider until something disrupts its production. Your gut has its own independent nervous system called the second brain. Your digestive system contains an independent network of approximately 500 million neurons called the enteric nervous system, often referred to as the second brain because of its remarkable autonomy. This neural network can operate entirely on its own, controlling the muscular contractions that move food through your digestive tract without requiring input from your central nervous system. The enteric nervous system uses more than 30 different neurotransmitters, including large quantities of serotonin, a chemical usually associated with mood regulation in the brain. This connection helps explain why digestive issues often accompany anxiety and depression, and why emotional states affect gut function so directly. The two-way communication between gut and brain travels primarily through the vagus nerve, carrying signals that influence everything from hunger and satiety to emotional states. Your gut neurons evolved over 500 million years ago, predating the development of the central nervous system as we know it. This ancient neural network continues to process information and control vital functions independently of conscious awareness. Your digestive system has more neurons than your spinal cord. The enteric nervous system embedded in the walls of your digestive tract contains approximately 500 million neurons, substantially more than the roughly 100 million neurons found in the spinal cord. This concentration of neural tissue is necessary to coordinate the complex muscular movements and secretory activities required for proper digestion. Every time you eat, your digestive system must respond with precisely timed waves of contraction, carefully regulated enzyme release, and appropriate blood flow adjustments. This neural network monitors gut contents through mechanical and chemical sensors, adjusting its response based on what you have eaten and how your body needs to process it. The enteric neurons work largely independently, but maintain constant communication with the brain through the vagus nerve and other pathways. Damage to this gut nervous system can cause severe digestive problems, even when the brain and spinal cord function normally. The remarkable neural density of your digestive tract reflects the evolutionary importance of efficiently extracting nutrition from food. Your muscles are always slightly contracted even at rest. Even when you are completely relaxed, your muscles maintain a baseline level of tension called muscle tone that keeps you ready for movement. This constant low-level contraction is controlled automatically by your nervous system, which continuously sends signals to muscle fibers to maintain a state of partial readiness. Muscle tone is what keeps your body stable and upright, preventing you from collapsing into a formless heap when you try to stand or sit. During sleep, muscle tone decreases significantly, which is why people can look so relaxed and sometimes drool or let their mouths fall open. Conditions that affect the nervous system can lead to either abnormally high muscle tone, causing stiffness and spasticity, or abnormally low tone, causing weakness and floppiness. Physical therapists and doctors assess muscle tone as an important indicator of nervous system function and overall health. This ever-present tension in your muscles represents the hidden effort your body expends every moment just to maintain its basic structure. You use over 30 muscles just to smile. The simple act of smiling engages between 30 and 50 muscles depending on the type and intensity of the expression, coordinating activity across your entire face. A genuine smile, called a Duchenne smile after the French neurologist who studied it, involves not just the muscles around your mouth but also the muscles around your eyes that create characteristic crow's feet wrinkles. Each side of your face controls its muscles somewhat independently, which is why asymmetrical smiles are common and can sometimes indicate underlying neurological differences. The brain areas that control voluntary smiling differ from those that control spontaneous emotional smiles, which explains why posed smiles often look different from genuine ones. Facial muscles are unusual because many attach to skin rather than to bone, allowing the subtle movements that create the full range of human expressions. Children learn to smile very early, with social smiles appearing within the first few months of life as the brain develops facial control. The coordination required to produce even a simple smile represents remarkable neural processing that most people accomplish without any conscious effort. The longest muscle in your body runs from hip to knee. The sartorius muscle holds the distinction of being the longest muscle in the human body. running diagonally across the front of your thigh from the hip bone to the inside of the knee. Despite its impressive length of approximately 60 centimeters in an average adult, the sartorius is actually a relatively thin, strap-like muscle rather than a bulky powerhouse. Its name comes from the Latin word for tailor, reflecting the cross-legged sitting position that tailors traditionally used, and which this muscle helps accomplish through hip flexion and rotation. The sartorius assists in several complex movements, including bending the hip, rotating the thigh outward, and flexing the knee, making it important for activities like walking, running, and climbing stairs. Because of its length and superficial position, the sartorius is vulnerable to strain injuries, particularly in athletes who perform explosive movements. The muscle's diagonal orientation across the thigh helps stabilize both the hip and knee joints during movement. This elegant strip of muscle demonstrates how length and position can be as important as raw power in the body's mechanical system. The Achilles tendon can handle over 1,000 pounds of force. The Achilles tendon connecting your calf muscles to your heel bone is the strongest tendon in the human body, capable of withstanding forces exceeding 1,000 pounds during intense activities like jumping or sprinting. This remarkable strength is necessary because the tendon must handle the enormous loads generated during activities where your entire body weight is accelerated and decelerated through your feet. Despite its impressive strength, the Achilles tendon is vulnerable to injury, particularly in middle-aged athletes whose tendons have begun to lose elasticity, but who continue to demand peak performance. A complete Achilles tendon rupture is often described as feeling like being kicked in the back of the leg, and the injury can be heard as an audible pop. The tendon takes its name from the Greek hero Achilles, whose only vulnerable spot, according to legend, was his heel, where his mother held him while dipping him in the river Styx. Regular stretching and gradual increases in exercise intensity can help maintain tendon health and reduce injury risk. The engineering of this single tendon demonstrates the remarkable forces your body routinely handles and controls. Muscles remember previous workouts for months. Muscle memory is a real phenomenon, in which previously trained muscles regain strength and size more quickly when training resumes after a break, even after months or years of inactivity. This memory is stored not in the muscles themselves, but in structural changes within muscle cells, including additional nuclei that remain even when the muscle shrinks from disuse. When you first build muscle through training, your muscle fibers recruit extra nuclei from nearby satellite cells to support the increased protein production needed for growth. These additional nuclei persist even during periods of detraining when muscle size decreases, providing a ready infrastructure for rapid rebuilding when training resumes. Research suggests this muscle memory can last for many years, which explains why former athletes can regain fitness more quickly than people who have never trained. The phenomenon provides encouragement to anyone returning to exercise after a break, knowing their previous efforts were not entirely lost. This cellular memory represents an evolutionary adaptation that allowed our ancestors to quickly regain physical capacity when conditions demanded renewed activity. Your muscles generate tiny electrical fields even at rest. Every muscle in your body produces small electrical signals as part of its normal function, and these bioelectrical fields can be detected and measured using a technique called electromyography. These signals originate from the depolarization waves that travel along muscle fibers when they receive the command to contract from motor neurons. Even at rest, small numbers of muscle fibers are contracting to maintain muscle tone, producing a continuous low-level electrical hum that specialized equipment can detect. Doctors use electromyography to diagnose muscle and nerve disorders, identifying abnormal patterns that indicate conditions like muscular dystrophy, nerve damage, or motor neuron disease. The electrical activity of muscles increases dramatically during movement, with the signal strength corresponding to the force of contraction. Athletes and researchers use these signals to study muscle activation patterns and optimize training techniques. Your body is constantly generating this electrical activity, making you a walking bioelectrical generator producing tiny voltages throughout your musculature. Each of your kidneys contains around 1 million tiny filters. Each kidney contains approximately 1 million microscopic filtering units called nephrons, which together process about 180 liters of blood every day. These tiny structures work continuously to filter waste products, excess water, and unwanted substances from your blood, while retaining essential proteins and blood cells. The nephrons accomplish this through a sophisticated two-stage process of filtration and selective reabsorption that recovers most of the water and useful molecules while concentrating waste into urine. Despite this enormous filtering capacity, you lose just 1 to 2 liters of water per day as urine, with the rest being recycled back into your bloodstream. The nephrons are remarkably efficient but cannot regenerate once damaged, which is why chronic kidney disease is so serious and often progressive. Each nephron contains a tuft of tiny blood vessels called a glomerulus, surrounded by a capsule that collects the filtered fluid. This million-unit army of microscopic filters works silently every second of your life to maintain the chemical balance your cells require for survival. The liver performs over 500 separate functions. Your liver is the largest internal organ and the most metabolically versatile, performing more than 500 different functions essential for survival. This chemical factory produces bile for fat digestion, synthesizes proteins needed for blood clotting, processes nearly everything you eat or drink, and stores vitamins and minerals for later use. The liver also detoxifies harmful substances including alcohol, drugs, and metabolic waste products, converting them into less harmful compounds that can be eliminated. Unlike most organs, the liver has remarkable regenerative capacity and can regrow to its full size from as little as 25% of its original mass. Blood supply to the liver comes from 2 sources, receiving both oxygen-rich blood from the hepatic artery and nutrient-rich blood from the intestines through the portal vein. The organ processes approximately 1.5 liters of blood every minute, making it one of the most metabolically active tissues in your body. This extraordinary multitasking organ truly deserves its reputation as the body's master chemist. The liver can detoxify almost any natural poison up to a point. Your liver has evolved sophisticated enzyme systems capable of neutralizing a vast array of natural toxins that might enter your body through food, water, or the environment. These detoxification pathways work through a series of chemical reactions that modify toxic molecules, making them water-soluble so they can be excreted through bile or urine. The liver's ability to handle toxins is impressive, but not unlimited, and overwhelming the organ with excessive alcohol, certain medications, or other poisons can cause serious damage. Some toxins, like those found in certain mushrooms, specifically target liver cells and can cause fatal damage, despite the organ's defenses. The detoxification capacity of the liver varies between individuals based on genetics, age, nutritional status, and the health of the liver tissue itself. Many medications require careful dosing, precisely because the liver's ability to process them determines how much can safely circulate in the bloodstream. This remarkable chemical defense system represents millions of years of evolution against the toxic challenges faced by our ancestors. You can survive without a large portion of your internal organs. The human body contains remarkable redundancy that allows you to survive and function reasonably well without significant portions of many internal organs. You can live with only 1 kidney, 1 lung, no spleen, no gallbladder, and large portions of your liver, stomach, and intestines removed. This biological spare capacity exists because our organs evolved to handle stress and damage while maintaining function. The liver can regenerate from just a quarter of its original mass, and a single kidney can filter blood effectively enough to maintain health. Some organs, like the spleen and appendix, are entirely dispensable, having functions that other tissues can compensate for, or that are no longer essential for modern humans. However, quality of life may be reduced, and certain precautions become necessary when organs are removed or reduced. This inherent redundancy has allowed humans to survive injuries and diseases that might otherwise be fatal, providing biological insurance against the unpredictable challenges of life. Your skin is the largest organ in your body. If stretched out flat, an adult's skin would cover approximately 2 square meters, making it by far the largest organ in the human body by surface area and weight. This remarkable organ serves as your primary barrier against the outside world, preventing water loss, blocking pathogens, and protecting internal tissues from physical damage. The skin consists of 3 main layers, with the epidermis on top providing waterproofing, the dermis beneath containing blood vessels and nerves, and the hypodermis below offering insulation and cushioning. Your skin is constantly renewing itself, shedding millions of dead cells every day, and replacing them from below in a continuous cycle that completely renews the epidermis roughly every month. The skin also plays crucial roles in temperature regulation through sweating and blood flow control, sensation through millions of nerve endings, and vitamin D production through exposure to sunlight. As the visible boundary of your body, skin has profound social and psychological significance across cultures. This living envelope protects everything inside you, while providing your primary interface with the physical world. Fingernails grow faster on your dominant hand. Fingernails on your dominant hand tend to grow slightly faster than those on your non-dominant hand, a difference thought to result from increased blood flow due to more frequent use. The greater mechanical stimulation and minor trauma from regular use appears to trigger slightly more rapid nail matrix activity. Fingernails generally grow about 3 to 4 millimeters per month, though this rate varies with age, nutrition, health status, and even the season. Nails grow faster during summer and in younger people, and pregnancy often accelerates nail growth due to hormonal changes. The middle fingernail typically grows fastest, possibly because it is the longest finger and receives the most use and blood supply. Toenails grow considerably slower than fingernails, typically taking 12 to 18 months to completely replace, compared to about 6 months for fingernails. These growth rate differences demonstrate how even small variations in blood flow and stimulation can affect cell division and tissue production. A single hair can hold about 100 grams of weight. Despite being just a fraction of a millimeter thick, a single human hair can support roughly 100 grams of weight before breaking, about the weight of a small apple. This impressive strength comes from the structure of hair, which consists primarily of keratin proteins, arranged in tightly coiled chains, surrounded by overlapping cuticle scales. When you multiply this strength by the approximately 100,000 hairs on an average head, the combined tensile strength could theoretically support the weight of 2 elephants. Hair's strength-to-weight ratio rivals that of many industrial materials, which is why engineers have studied its structure for biomimetic applications. The elasticity of hair allows it to stretch up to 30% of its length before breaking, providing resilience against normal brushing and styling stresses. Hair strength varies with color, diameter, and condition, with thicker Asian hair generally being strongest and fine blonde hair being more delicate. This remarkable structural material grows continuously from follicles embedded in your scalp throughout your life. Hair and nails do not keep growing after death. Contrary to popular belief, hair and nails do not continue growing after death, though they may appear longer, due to skin dehydration and retraction. When the heart stops beating, cells lose their oxygen supply, and the energy-requiring processes of cell division and protein synthesis cease within hours. The illusion of postmortem growth occurs because the surrounding skin dries out and shrinks, pulling back from the nail beds and receding along the hair shafts to expose more of what was previously hidden. This optical illusion has persisted in folklore for centuries, contributing to macabre stories about corpses with grotesquely long nails and hair. Understanding that growth requires active metabolism helps explain why this myth cannot be true, as the complex machinery of cell division needs constant energy input. The same dehydration that creates this illusion also causes facial features to appear more gaunt and pronounces bony structures. This medical myth serves as an interesting reminder of how appearances can deceive us about the actual processes occurring in biological systems. There are more nerve endings in your fingertips than almost anywhere else. Your fingertips contain one of the highest concentrations of touch receptors anywhere in your body, with approximately 2,500 nerve endings packed into each square centimeter of skin. This extraordinary sensory density allows you to detect textures, temperatures, and pressures with remarkable precision, enabling tasks from reading Braille to threading a needle. The high concentration of receptors in the fingertips is reflected in the large area of the brain's sensory cortex devoted to processing touch information from the hands. If the body were drawn in proportion to its sensory representation in the brain, the hands, and especially the fingertips, would be enormously enlarged compared to the trunk and legs. 4 main types of mechanoreceptors in the fingertips detect different aspects of touch, including light pressure, vibration, sustained pressure, and skin stretch. The sensitivity of your fingertips can detect surface variations as small as a few micrometers, smaller than individual cells. This exquisite tactile precision has been essential for human tool use and manipulation throughout our evolutionary history. Your DNA unravels and rewraps millions of times per day. Every time a cell needs to read the instructions contained in its DNA for protein production or cell division, the tightly wound double helix must unravel and then rewind after the information is copied. This unwinding and rewinding occurs millions of times daily across the trillions of cells in your body, representing an enormous amount of molecular activity happening at every moment. Specialized enzymes called helicases unwind the DNA strands, while other proteins stabilize the separated strands, and yet others handle the rewinding process. The process must be precisely controlled, because errors in unwinding or rewinding can lead to mutations, broken strands, or genes being expressed at the wrong times. This constant unwinding is necessary because DNA is stored in an incredibly compact form, with roughly 2 meters of genetic material packed into each cell nucleus. The compaction is achieved through wrapping DNA around protein spools called histones, which must temporarily release their grip when genes need to be accessed. This perpetual dance of unwrapping and rewrapping is fundamental to every process in your living body. Every cell contains around 2 meters of DNA. If you could unwind and stretch out the DNA contained in a single human cell, it would extend approximately 2 meters in length, yet this enormous molecule is packed into a nucleus, only about 6 micrometers across. This remarkable feat of biological packaging is accomplished through multiple levels of coiling and folding, with DNA wrapping around histone proteins like thread around tiny spools. These nucleosomes then coil into larger structures that fold again into the chromosomes visible during cell division. If all the DNA in your body were uncoiled and laid end to end, it would stretch to the sun and back multiple times, covering a distance of billions of kilometers. Despite this extraordinary length, DNA is incredibly thin, measuring only about 2 nanometers in diameter. The information contained in this thin strand includes approximately 3 billion base pairs coding for an estimated 20,000 genes plus vast regulatory regions. This microscopic library contains all the instructions needed to build and maintain your entire body. Your body is made of stardust forged in ancient stellar explosions. Nearly every atom in your body except hydrogen was created inside dying stars through the process of stellar nucleosynthesis billions of years ago. The carbon that forms the backbone of your molecules, the oxygen you breathe, the calcium in your bones, and the iron in your blood were all forged in the nuclear furnaces of ancient stars. When these massive stars exploded as supernovae, they scattered these heavy elements across space, where they eventually coalesced into new star systems, including our own solar system and planet. The hydrogen in your body is even older, formed shortly after the Big Bang itself some 13.8 billion years ago. You are literally made of the same matter that composes stars, planets, and the cosmos itself, temporarily arranged in the pattern that makes you uniquely you. This remarkable connection between your physical form and the universe is not poetic metaphor, but scientific fact, supported by spectroscopic analysis and nuclear physics. Every atom in your body has a cosmic history stretching back billions of years before your birth. There are about 37 trillion cells in the human body. The human body contains approximately 37 trillion cells, though estimates vary depending on the counting methodology and individual body size. This number is so vast that if you tried to count every cell at a rate of 1 per second, it would take over 1 million years to complete the task. These cells come in more than 200 different types, ranging from tiny red blood cells just 7 micrometers across to nerve cells with axons stretching 1 meter or more. Each cell except red blood cells contains a complete copy of your genome, yet different cells express different genes to create their specialized structures and functions. Cells vary enormously in their lifespans, with some intestinal lining cells surviving only a few days, while certain neurons last an entire lifetime. The coordination required to keep 37 trillion cells working together harmoniously represents one of the most complex organizational challenges in the known universe. This vast cellular community is continuously communicating, dividing, dying, and being replaced in the ongoing process that keeps you alive. A fetus can heal wounds without leaving scars. In the first 2 trimesters of pregnancy, a human fetus can heal wounds without any scarring whatsoever, regenerating tissue that is indistinguishable from the original uninjured tissue. This remarkable ability is lost before birth, and by the 3rd trimester, healing begins to produce scars similar to those seen in children and adults. Research into fetal wound healing has identified several factors that differ from adult healing, including reduced inflammation, different growth factor profiles, and unique properties of fetal skin cells. The fetal wound healing environment is also unique, with the amniotic fluid possibly providing factors that support regenerative rather than scarring repair. Scientists are intensively studying this phenomenon, hoping to develop treatments that could help adults heal without scarring after surgery or injury. Burns, surgical incisions, and traumatic wounds could potentially heal perfectly if we could recreate the conditions present in early fetal development. Understanding this natural regenerative ability represents one of the most exciting frontiers in wound healing research. More than half your body is not human but microbial. By cell count, more than half the cells in and on your body are not human cells but microorganisms, including bacteria, archaea, fungi, and viruses that collectively form your microbiome. While human cells are typically much larger than bacterial cells, meaning you are still mostly human by mass, the sheer number of microbial cells rivals or exceeds your own cell population. These microorganisms colonize virtually every surface of your body, from skin to gut to respiratory tract, forming complex communities adapted to each unique environment. Far from being harmful parasites, most of these microbes provide essential services, including digestion, vitamin production, immune system training, and protection against pathogenic invaders. The diversity of your microbiome develops from birth onward, influenced by delivery method, breastfeeding, diet, environment, and the people and animals you contact. Disruption of this microbial community through antibiotics or illness can have significant effects on health that extend far beyond the digestive system. You are not so much an individual organism as a collaborative ecosystem of human and microbial life. Your gut bacteria weigh about 3 pounds. The bacterial community in your digestive tract collectively weighs between 1 and 2 kilograms, roughly the same as your brain, making it one of the heaviest microbial communities on Earth. This substantial mass represents trillions of individual organisms from hundreds of different species, each specialized for its particular niche within your intestinal ecosystem. The gut microbiome is densest in the large intestine, where bacteria ferment dietary fiber and other materials that escape digestion in the small intestine. These bacteria produce vitamins including K and several B vitamins, short-chain fatty acids that nourish intestinal cells, and signaling molecules that affect metabolism and immune function. The composition of gut bacteria varies dramatically between individuals and is influenced by diet, with different foods selecting for different bacterial populations. This bacterial mass is not static but constantly changing, with vast numbers of bacteria dying and being replaced, while overall population stays relatively stable. Your gut microbiome represents a living organ of sorts, contributing metabolic capabilities that your human cells alone could not provide. Your microbiome is unique to you like a fingerprint. The specific combination of microbial species in your body is so individual that researchers can identify people from their bacterial signatures alone, much like fingerprint identification. This uniqueness develops from the first moments of life and is shaped by countless factors including genetics, diet, living environment, pets, family members, and health history. Even identical twins living in the same household develop different microbiome compositions, showing that environmental influences eventually distinguish even genetically identical individuals. The skin microbiome varies dramatically across different body regions, with the oily skin of the forehead harboring different species than the dry skin of the forearms or the moist environment between toes. Forensic scientists are exploring microbiome analysis as a tool for identifying individuals from bacterial traces left at crime scenes. The stability of your core microbiome is remarkable, with your unique signature persisting even after temporary disruptions from illness or antibiotics. This microbial fingerprint represents a biological identity card written by your life experiences and environment. Gut bacteria influence your mood and brain chemistry. The bacteria in your digestive system produce neurotransmitters including serotonin, dopamine, and gamma-aminobutyric acid that can influence brain function and emotional states through what scientists call the gut-brain axis. Approximately 90% of the body's serotonin, a neurotransmitter crucial for mood regulation, is actually produced in the gut rather than the brain. Signals travel between gut and brain through the vagus nerve, the immune system, and molecules that bacteria produce that can cross the blood-brain barrier. Studies in both animals and humans have shown that changes in gut bacteria composition can affect anxiety, depression, stress response, and even social behavior. Probiotic supplements and dietary changes that alter gut bacteria have shown promise in improving mood and reducing anxiety symptoms in clinical trials. The emerging field of psychobiotics explores how manipulating gut bacteria might treat psychiatric conditions alongside or instead of traditional medications. This connection between your intestinal passengers and your mental state reveals that your mood may depend partly on the health of your microbial community. Goosebumps are a leftover trait from when humans had more body hair. When you feel cold or experience strong emotions, tiny muscles attached to your hair follicles contract, pulling each hair upright and creating the bumpy skin texture we call goosebumps. In our hairier ancestors, this response served practical purposes, with raised fur trapping a layer of insulating air to provide warmth, or making the animal appear larger and more threatening to predators. In modern humans with our relatively sparse body hair, this vestigial response serves little practical purpose, yet persists as an automatic reaction controlled by the sympathetic nervous system. The same fight or flight response that raises your hair also increases heart rate, dilates pupils, and redirects blood flow to muscles, preparing your body for action. Goosebumps triggered by emotional experiences like inspiring music or moving stories may reflect the overlap between the fear response and other intense emotional states. The tiny erector pili muscles that create goosebumps are found throughout your skin, except on the palms of your hands and soles of your feet. This vestigial response provides a visible reminder of our evolutionary connection to other mammals. The human embryo has a tail for a brief period. During the 4th and 5th weeks of embryonic development, every human embryo develops a visible tail-like structure containing 10 to 12 vertebrae that extends beyond the developing legs. This tail normally regresses through programmed cell death about 3 weeks later, with its vertebrae either fusing or being absorbed, until only the vestigial coccyx remains as the adult tailbone. In rare cases, the regression process fails to complete, and babies are born with small tails that can be surgically removed without difficulty. The embryonic tail is just one of many structures that appear briefly during human development and reflect our evolutionary ancestry, much like the gill-like pharyngeal arches that later develop into parts of the face and throat. These temporary embryonic features form because the same developmental genes that build tails in other mammals are still present in our DNA and become active during early development. The regression of the tail is controlled by genes that evolved in our tailless primate ancestors millions of years ago. This developmental journey recapitulates aspects of our evolutionary history in miniature. You have a reflex that prevents you from biting your own tongue. Your tongue is protected from being bitten during chewing by a sophisticated reflex arc that automatically moves your tongue away from your closing teeth faster than conscious thought could manage. Sensors in your tongue detect the approach of your teeth and trigger an immediate motor response that repositions the tongue before injury can occur. This reflex works so well that despite the constantly moving danger of your teeth, most people bite their tongues only occasionally, and usually only when the reflex is disrupted by distraction, surprise, or certain neurological conditions. The tongue is remarkably dexterous, containing 8 muscles that work together to perform the complex movements required for speech, swallowing, and manipulating food during chewing. The speed of the protective reflex is essential because jaw-closing muscles are powerful enough to generate forces that could easily sever the tongue if it were caught between the molars. When tongue bites do occur, they are usually partial and heal quickly due to the excellent blood supply and regenerative capacity of oral tissues. This automatic protective system demonstrates the sophisticated integration of sensation and movement in even the most basic functions. People with synesthesia can taste sounds or see colors in music. Synesthesia is a neurological phenomenon in which stimulation of one sensory pathway automatically triggers experiences in another, causing people to taste words, see colors when hearing music, or perceive letters as having inherent colors. This unusual crossing of sensory wires occurs in approximately 4% of the population and appears to have a genetic component, often running in families. The most common form involves perceiving letters and numbers as having specific colors, but dozens of different types have been documented, including rare forms where people feel physical sensations on their body when observing others being touched. Brain imaging studies show that synesthetes have increased connections between sensory areas of the brain that are normally more separate, explaining why stimulation in one area activates another. Far from being a disorder, synesthesia is often associated with enhanced memory, creativity, and artistic ability, with many famous artists and composers having reported synesthetic experiences. The phenomenon may represent a failure of the normal developmental pruning that separates sensory circuits in early childhood. Synesthesia provides a fascinating window into the flexibility and variation possible in human perception. The human body emits faint bioluminescent light invisible to the eye. Your body continuously emits extremely faint light through a process called ultra-weak photon emission, or biophoton emission, though this glow is about 1,000 times too dim for human eyes to detect. This biological light results from chemical reactions occurring in living cells, particularly reactions involving free radicals and the oxidation of molecules like lipids and proteins. The emission intensity varies throughout the day, following circadian rhythms, with the highest glow occurring in the late afternoon around the mouth and cheeks. Researchers using ultra-sensitive cameras in completely dark conditions have mapped this ghostly glow across the human body, finding it strongest around the face. Unlike bioluminescence in fireflies or deep-sea creatures, which uses dedicated light-producing chemistry, human biophoton emission is a byproduct of normal metabolism. Some scientists speculate these photons might play a role in cellular communication, though this remains under investigation. This invisible radiance means you are technically glowing right now, emitting light that only the most sensitive scientific instruments can detect. Your body contains trace amounts of gold. The average adult human body contains approximately 0.2 milligrams of gold, mostly dissolved in blood where it circulates at concentrations too low to measure without specialized equipment. This gold enters your body from trace amounts in food, water, and the environment, and is excreted over time so that the body maintains a roughly constant level. At current gold prices, the gold in your body would be worth less than a penny, so there is no fortune to be extracted from human tissue. Beyond gold, your body contains many other surprising elements, including traces of uranium, thorium, and mercury, accumulated from environmental sources. The gold in your bloodstream is being continuously filtered through your kidneys and recycled, with only tiny amounts lost in urine each day. While the amount is far too small to have any physiological function, the fact that you contain gold demonstrates how elements circulate through living systems from the environment. You carry within you trace amounts of nearly every naturally occurring element on Earth. Your body contains enough fat to make 7 bars of soap. The average adult carries enough body fat to produce approximately 7 standard-sized bars of soap, based on the historical process of making soap from animal fats through saponification with lye. This calculation assumes typical body fat percentages, with the actual number varying significantly based on individual composition. Body fat, primarily composed of triglycerides, has the same fundamental chemistry as the fats historically used in soap making from animals like pigs and cattle. Beyond its potential soap-making capacity, body fat serves crucial functions, including energy storage, insulation, organ cushioning, and hormone production. Fat tissue is metabolically active, producing hormones and chemical signals that regulate appetite, inflammation, and reproductive function. The distribution of fat across your body changes with age and hormonal status, with visceral fat around internal organs being more metabolically active and potentially harmful than subcutaneous fat under the skin. This unusual metric for measuring body fat content provides a concrete illustration of the material resources stored within the human form. Some of your cells are older than your bones. While most cells in your body are replaced regularly, certain neurons in your brain and inner ear are as old as you are, having been present since birth and persisting throughout your life without replacement. The lens cells at the center of your eye are similarly ancient, formed during embryonic development and never replaced thereafter. These long-lived cells must maintain themselves through continuous repair and maintenance rather than replacement, which is one reason why age-related changes affect these tissues particularly. In contrast, your bone cells are constantly being renewed as bone is remodeled throughout life, meaning the bone you have now is actually younger than you might expect. Red blood cells live only about 120 days, intestinal lining cells survive just a few days, and skin cells are replaced every few weeks. This patchwork of cellular ages means your body contains elements from throughout your entire lifespan, coexisting with cells created just moments ago. You are simultaneously ancient and continuously renewed in different parts of your being. You only perceive a tiny fraction of the sensory input your body receives. Your brain receives billions of bits of sensory information every second, but consciously processes only a minuscule fraction, filtering out most input before it ever reaches awareness. This filtering is essential because conscious processing is too slow and limited to handle the full flood of data from your senses, so your brain selects what seems most relevant and ignores the rest. You are not aware of the constant pressure of clothing on your skin, the ambient sounds of your environment, or the position of your tongue in your mouth until your attention is drawn to them. The information that reaches consciousness has been heavily processed, interpreted, and sometimes altered by your brain's expectations and prior experiences. Optical illusions and perceptual phenomena demonstrate how much construction occurs between raw sensory input and conscious experience. Your brain essentially creates a useful model of reality rather than providing direct access to objective truth. What you perceive as the world is actually a carefully edited story your brain tells based on limited samples of the information available. The brain uses chemical gradients to organize thought patterns. Your brain organizes its activity partly through concentration gradients of neurotransmitters and neuromodulators that bathe different regions and influence local neural activity. These chemical gradients create functional zones where neurons respond differently to the same inputs depending on the local chemical environment. Neurotransmitters like dopamine, serotonin, and norepinephrine are released in patterns that shift with attention, arousal, reward, and emotional state, modulating how information flows through neural circuits. This chemical landscape changes continuously throughout the day, contributing to variations in mood, alertness, and cognitive performance. Sleep involves dramatic shifts in brain chemistry, with different neurotransmitter patterns characterizing different sleep stages. The brain's chemistry can be influenced by external factors, including food, exercise, light exposure, and social interaction, explaining why these factors affect mental function. Understanding these chemical gradients is key to understanding both normal brain function and the mechanisms of psychiatric medications that work by altering neurotransmitter levels. Your face keeps changing even after your bones stop developing. While your facial bones largely complete their growth by your early 20s, your face continues to change throughout life due to ongoing bone remodeling, fat redistribution, and changes in soft tissue. The bones of your face actually recede slightly with age, with the eye sockets enlarging and the jaw losing volume, contributing to the appearance of aging beyond what skin changes alone would cause. Fat pads in the face shift downward over decades, moving from areas around the cheeks to accumulate along the jaw and under the chin. The muscles of facial expression can also change with habitual use, with frequently used muscles becoming more prominent while others weaken. Skin changes, including loss of collagen and elastin, compound these deeper structural changes to produce the familiar progression of facial aging. Identical twins who age in different environments develop noticeably different facial appearances over time, demonstrating how much lifestyle and environment contribute to these changes. Your face is never truly finished, continuing to reshape itself throughout your entire lifetime. Your bones constantly replace old calcium with new. Your skeleton is continuously remodeling itself through a carefully balanced process of bone breakdown and rebuilding, completely replacing itself roughly every 10 years in adults. Specialized cells called osteoclasts dissolve old bone tissue, releasing calcium and other minerals back into the bloodstream, while creating small cavities in the bone. Other cells called osteoblasts then fill these cavities with new bone tissue, incorporating fresh calcium from your diet and bloodstream. This constant turnover allows bones to repair microdamage, adapt their structure to mechanical stress, and serve as a reservoir for calcium needed by other body systems. When dietary calcium is insufficient, more bone is broken down than built up, leading to gradual weakening and conditions like osteoporosis. Weight-bearing exercise stimulates bone building by creating the stress signals that prompt osteoblasts to strengthen areas of high mechanical demand. Your seemingly solid skeleton is actually a dynamic tissue in constant flux, quietly rebuilding itself while you sleep each night. The body rebuilds itself so completely that you are always becoming new. The atoms and molecules that compose your body are constantly being replaced through metabolic processes, so that you are materially a different person than you were months or years ago. Red blood cells last about 4 months, skin cells about 2 to 3 weeks, and the lining of your digestive tract replaces itself every few days. Even your skeleton, which seems so permanent, replaces itself roughly every decade through the gradual turnover of bone tissue. Some estimates suggest that over 98% of the atoms in your body are replaced each year, with only certain long-lived neurons and lens cells persisting unchanged throughout life. What remains constant is not the matter itself, but the pattern, the organization of information that makes you distinctly you across time. This continuous replacement means you are less a fixed object than a persistent pattern, like a wave that maintains its form while the water constantly changes. In a very real sense, the body you have now is almost entirely different from the body you had a decade ago. And now the quiet wonders of the human body begin to gently fade into the stillness of the night. You have wandered through over 100 peaceful truths. From the 25 million cells created every second to the 100 trillion connections within your brain. From the ancient stardust in your blood to the silent electricity humming through your nerves. You have explored the hidden rhythms of your heart, walked through the vast networks of your digestive system, and discovered how your bones rebuild themselves in silence while you sleep. You are part of this story. Not just an observer, but the story itself. Every cell, every beat, every breath is a chapter in the book of you. The atoms that make your body have been part of stars, oceans, and countless living things before finding their way to you. If your eyes feel heavy now, let them close. Let the rhythm of your breath become steady, like the gentle pulse of your heart. Like the patient work of cells renewing themselves in the darkness. Tonight, as you rest, your body continues its quiet work. Bones are rebuilding. Cells are dividing. Immune cells are patrolling. Somewhere deep within you, memories are being consolidated, and dreams are preparing to unfold. You are a small, warm universe unto yourself, peacefully resting among the wonders of existence. Sleep well, sweet dreams, and may you carry with you the quiet knowledge that you are a miracle of biology, chemistry, and physics. A collection of stardust that somehow learned to wonder about itself. Thank you for joining me on this journey through the wonders of the human body. If you enjoyed this video, please subscribe to Drowsy Discovery for more soothing explorations of science and our remarkable universe. There are more wonders waiting, more secrets hidden in cells and synapses, more mysteries to drift through on quiet evenings. Rest well, and may the gentle rhythm of your own heartbeat keep you company through the night. This has been Drowsy Discovery. Good night.